know, but I didn't bring it. So over here, there's the 50, I can't say it all, but it's the 50 base pair ladder. So we're gonna count from the bottom, 50, 100, 150. The bright one seems to be 200. Then we get 250, 300. I guess you can see the 350, maybe you can label that. Make sure it's clear that these units are in base pairs. And I think those seem to be labeled correctly because when we added up the entire amplimer, I think we said it's 272 or 273 if we're talking about the mutation, which is an extra A at the position of that red star. So 272 or 3 certainly seems consistent with that signal there between the 250 and the 300. Let's see, then we had some controls. So you might label this as your uncut amplimer. This one was genotypically normal. This one was heterozygous. This one was the 2J mutation. And I think we can see everything that the diagram suggests that we should. For the wild type, both of those highlighted sequences are the correct target sequence for the MLY1 enzyme. And so we get the 3 fragments. That first fragment is 122. The second fragment is the 69 base pairs for the wild type, and the last fragment should be 81. And I think those make sense. 81 is here, 69 is there, and that's consistent with our size standard. And the 122 should be right in between the 100 and the 150. So those sizes seem right. The brightness, brightest, middle brightness, least brightness, that's sort of consistent. Our original amplimer was presumably here, and I think it looks like it cut to completion. We've got the 3 signals that we expected for the wild type control. So really that is a control. The 2J should lack that highlighted sequence because of the addition of an A. That is no longer the target site for MLY1, but this one is. So we should get the overall fragment cutting here only. And so that should be 122 plus 70, so 192. So that's what that seems to be. And then the smaller fragment is 81, exactly the same as that one there. So just the 2 bands when the only template is the mutant sequence, which lacks the cut site further up in the diagram. The heterozygote should have both. It should have the mutant signal here and here, as well as the wild type signal here, here, and here. And since, this is half as bright as that because it's half as many templates. This one's so dim that we can barely see it. This one's nice and bright because we get it from both the wild type and from the mutant. And so that one does show up quite strongly. But I think the little one really is there in these heterozygotes. So then everybody else, now that the controls have all given us pretty much exactly what we thought we should see, presumably the whole experiment has gone to plan. Those are our 13 kids. And you may want to be talking about these kids, so giving them each a name, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, might be helpful to you. They are all the result of that test cross that we did with a wild type CW2J heterozygote crossed back to the normal parent. So we're expecting all the kids to get plus from the normal parent. Half the kids will get plus from the F1 parent, half the kids will get CW2J. So we're expecting half and half, a 1-to-1 ratio. So did we get that? It looks like we have 1, 2, 3, 4, 5, 6, 7, 8 in the wild type category. And the rest seem to be heterozygous. 1, 2, 3, 4, 5 of the heterozygous variety. So it's not exactly 50-50, but you don't expect to flip a coin and get exactly 50-50. I don't think that looks different from 50-50. 6.5 would be perfect, so we're kind of close to that. Certainly, I think that's enough sightings of this heterozygous genotype. I'm pretty convinced that we had this guy, the parent, the heterozygous parent, was correctly identified. And that was one of the things that we were kind of concerned about. We certainly have identified 5 kids that seem to carry, if we've done everything right and haven't mixed up any samples, and those will be the ones that we can cross to the wild type again to make another 50-50 generation. And that sighting of a 50-50 generation next time around will confirm the genotype that we are proposing here. So it seems like if this is 1, 2, 3, number 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, those would be the ones that I might name as the ones that we suggest are carriers, and therefore should set a wedding date for sometime in the next 8 weeks. So yes, something above that line. That line kind of unites all of these columns. These are all the kids of a test cross like this. You got a lot of space there. You can probably fit something fairly descriptive. Or I think we might have written on the board yesterday, test cross, back cross, progeny. That means a group of kids. All right? Yeah.
Is there, probably a stupid question, but is there such a thing as being too descriptive when labeling the diagram? Because I know we have to make it understandable for somebody who hasn't done research.
You're making decisions, certainly. Sometimes I labeling the diagram, I don't like. I mean, some people will say, you'll see this in textbooks and stuff, and it's frustrating as hell. They'll just go 1, 2, 3, 4, 5, 6. They'll number the 17 lanes, and then you have to read the whole darn paper to find out what's in lane 7. I like it much better if I can tell, oh, here's my controls right here. And then I can kind of stop reading and figure out what's going on over there.
Okay.
So you have to decide what's helpful. Like on the last report that I just finished grading, a whole bunch of you just printed out the picture and said A, B, C, D. I'm like, well, how helpful is that? And I thought, well, maybe it's fine. Maybe if they tell me what A, B, C, and D are. But then I would read the legend and I'd find out that A meant to you the plate, the cells, the plasmid, the original sample. All that stuff was represented by A. And so A kind of lost all its meaning when, DNA is not equal to cell, is not equal to test tube, is not equal to plate. So that got to be a bit rough. So yeah, can you go too far? I suppose you can. Sometimes I look at it and I like to use this space. I certainly want to use this space. I got all this free space over here. What might I do? I might indicate again what I'm suggesting to be the sizes and base pairs of those bands.
Okay.
I'm thinking I might not label this guy because I'm going to have to draw on the picture, and it's not real convenient over here, and it's so far from this side I might not bother, but then again I might. Rather than force the reader to kind of figure out the sizes of these bands, I might fill them right in. Like for example, this is, we said 192? Certainly use units. We only see that when it's the CW2J allele. I mean, that's kind of what we're picking up on. We only see this. What did we say that was? 122?
Yeah.
We cite that exclusively when it's the wild type allele. And if I'm going to use plus to mean the wild type allele, I'm going to have to say so down in the legend. Plus represents the wild type allele of, maybe somewhere we have to say the real name of this gene is HEF-like 1. That's really what this whole report is about. That certainly is title worthy. Okay, so that kind of seems helpful. If this legend is all about determining which allele is which, the more things I can tell my reader that make it easy, the less things he's going to have to do. And now I got all this space underneath the picture, and sometimes I kind of want to use my space without driving anybody crazy. We are looking at a 3.5% Nusik gel. That's certainly true. You just don't want to write anything that's untrue. Untrue stuff is horribly unhelpful. Ambiguous stuff is not especially helpful. But untruths, that fake news thing, is a drag. So things that are true and are helpful. Yeah.
What is a backcross?
A backcross is when you take, well, you can define it narrowly, you can divide it widely. Here we have a heterozygote, and we're crossing back to one of the genotypes that produced that heterozygote. Sometimes we maybe wrongly assume that this was a wild type parent gave that and a mutant parent gave that, and now we're crossing back to either one of those, either the wild type parent or the mutant parent. Test cross is something we use, although that's specifically the back cross when you cross back to the recessive. This is like a little mini genetics class. If the term is confusing, you might just want to maybe show genetically what the cross is somewhere. Maybe over that line, maybe in the title, maybe right after the title. So what are the things that go in the title? Again, I kind of start with a list of things that seem important to me. You know, electrophoresis is not important, that's just a technique. What were we trying to do here? What does every title in biology have to have? The name of the species. So a bunch of you lost points last time because there was no mention of E. coli anywhere. So this time, this is certainly about mice. So our goal is to determine the genotypes for a small family of mice. That whole phrase probably belongs in the title. Well, what genotypes? Can we be more specific? We're looking at exactly one gene. We're looking at the HEF-like1 gene. Plus represents the wild-type allele of HEF-like1. CW2J represents the mutant allele of HEF-like1. So you define those symbols that you use. But I think HEF-like1, since it's the only gene we're really thinking about, needs to be in the title. Let's see. We generated amplimers using PCR. That's going to have to be said someplace. We cut those amplimers with a single endonuclease, so let's name it MLY1 endonuclease. Amplification of HEF-like1. Now I'm going to edit myself. Amplification and restriction endonuclease digestion. Endonuclease digestion of amplimers of the HEF-like1 gene reveals the genotypes of a small family of mice. Something like that. We're trying to learn their genotypes. HEF-like1's got to be in there somewhere where it makes sense. You've got to say the word mice. This was an amplimer that we digested with a nuclease. Maybe those can be worked in without making it terrible. But sometimes I keep track of a list of, because I evaluate you by looking for the use of specific vocabulary. So I kind of want to know what vocabulary do I want to show off that I know how to use. Some of it's going to make it to the title. Some of it's going to not make it to the title, but it has to be in the rest of the legend. Now, in that legend, a whole big section of that's going to have to be protocol. We started with genomic DNA. That's pretty important. Isolated from this small family of mice. We amplified those templates using a standard polymerase chain reaction. And on the protocol, now I'm going to look and see. We want some details about what went into those reactions. And I'm not real satisfied what I see written down here. There was a forward and reverse primer. You might want to jot this down. It is a fact that in the final reaction, they were at a concentration of 6 millimolar. That fact doesn't seem to show up on this worksheet, but that is a fact. Primers forward and reverse, at a concentration of 6 millimolar in the final reaction mixture. Certainly the Taq DNA polymerase, that's the enzyme that was in the reaction. Again, we'll say it was 1 unit per 12 microliter reaction. Template DNA, 0.05 micrograms of mouse genomic DNA is what was used. And that specific detail's not there, but it does turn out that when we're describing a protocol of an experiment, we kind of do want the reader to pick up amounts, volumes, concentrations like this, so that this reaction could be reestablished by someone who was trying to do that. Does it matter we had a premix? No. Doesn't matter. Does it matter that our tubes were orange or green? No, it doesn't matter. So the stuff that went into that reaction is important. On the last page, in gray, the 3 different steps of the procedure. Reactions were denatured at 95 degrees, 30 seconds, annealed at 68 degrees, 10 seconds, and extended at 72 degrees, 10 seconds. So in a sentence, I'm able to say what the characteristics of the 3 steps were, and naming them using the appropriate language. The denaturation step, the annealing step, the extension step. I've got those words and they're even underlined on the protocol. So I know some of you guys want to write that down like it was a little list, but in a sentence is what we put in a figure legend like that. So now that's all taken care of. Now what we produced, those products, we've been calling amplimers. Those amplimers were then, on the second protocol, they were digested by incubation with the MLY1 restriction endonuclease. 1 unit of the enzyme is what we use, and it does say that on the protocol here. We estimated that each reaction contained 0.25 micrograms of that amplified DNA that we produced in the PCR reaction. I guess you could say the total volume of the digestion reaction looks like 3, 6, 9, 12 microliters. And I think with that much information, the units of enzyme, the amount of target DNA, and the volume, I think we can figure it out. The buffer, its concentration and components are kind of disguised by the manufacturer. So, with a buffer, according to manufacturer's recommendations. Just itchy. Yeah.
So when you're looking at that diagram up there,
Yeah.
that whole thing is to have like 1 gene?
No. Excellent question. Excellent question. Yeah, and I don't know if you want to include this in your diagram. I guess you could, because you've got this picture on those things the protocols which are posted on Blackboard if you want it. I knew I wanted to point to it while I was talking about this. The blue bases in capital letters, that is exon 14, and I think the handout does tell you that. So this insertion that we have here is in the smack dab the middle of a coding region. So clearly this mutation, the insertion of a single A, is going to set off a frame shift. And I think we'll see this in detail when I do seminar next Friday. Not this Friday, because there is no class this Friday. The black is the intron before exon 14, so it should be, it's just a portion of that very large intron 13 to 14. And this black is the beginning of intron 14 to 15. And it's kind of neat because we said there's always a GT at the beginning, there's always an AG at the end of an intron, and the splice is always looking for those. There's always a track that is pyrimidine rich. There is a bunch of pyrimidines here. So this does show you one example of that exon-intron boundary kind of thing. These are the primers that were used at a concentration of 6 millimolar. Yeah, that may be something that you find useful. If it's not useful to the story you're telling, then leave it out. But if it is useful, you might want to make this figure part A. This might be figure part B. But don't show it if you don't want to discuss it necessarily.
And what are those 2 arrows?
That's the position where the MLY endonuclease cuts. It's a weird one. That target sequence is sort of kind of palindromic with an extra C in the center. Then it leans over. It doesn't care what these bases are, 1, 2, 3, 4, 5, and then it cuts the backbone off to one side. In fact, you can Google this. You can ask Siri, what do you know about MLY1? And she'll take you to New England Biolabs, and they'll tell you this is a weird endonuclease. Both parts are leaning over the same way. It's kind of tricky. In fact, you'll even see what Siri tells you, and you'll say, Tom's out of his mind. That's not what they say the target site is at all. They happen to describe the other strand for reasons I don't necessarily know why they made that choice. But that is exactly where the backbone is cut, both top and bottom. Someone was waving at me, no? So we're still going through protocol. So now we're going to talk about the digestion, times, temperatures, and then after digestion, digests were mixed with loading buffer. I think that's more descriptive than sample buffer. Sample buffer can be anything. Loading buffer is going to have the blue dye and the thick sucrose-y stuff. And then they would put on that 3.5% Nuciv agarose gel, electrophoresed at 140 volts for about an hour. Gels were stained with lithium bromide and photographed under ultraviolet light. And so we've gone chronologically through what we did, and I don't think you want that to take up more than like half the page, but you want to have times, temperatures, voltages, all that key stuff, and kind of leave some room for yourself to talk about what this means. So you'll describe your controls, and then you'll interpret your family. Maybe you'll mention you were expecting 50-50, and you got kind of 50-50, although not precisely. You were specifically looking for the carriers, and we've identified 5. I don't remember if they're boys or girls, but some sort of mix would be lovely. Anything we can do better next time? I don't know, maybe something will occur to you. In a way, I feel like we've done this experiment a couple of times, and we did it wrong several times, and so we stuck in the uncut, we stuck in sufficient numbers of controls. We did a bunch of things that we didn't do initially. Initially, we just worked with the 13 mice, and we're left scratching our heads at the end of it. Maybe we've kind of fixed a lot of the problems. Maybe you'll notice something as you go through it. Any questions that occur to you guys? Unfortunately, fortunately, we've got Friday off, we've got Monday off, so this is like due on Tuesday. So if you do have questions, the sooner you come up with those questions, the better. Email me, I'll do my darndest on the weekend to pay attention to you. But it is my birthday, so. Okay? Let me